So we are gathered today for the PhD defense of François Stockart, who realized his PhD work in the Laboratoire de Psychologie et de Proposition at Université Grenoble-Alpes. And he realized this thesis at École Doctorale EDIS in the specialty cognitive sciences, cognitive psychology and neurocognition. So this work was supervised by Nathan Febvre, who is a member of the LPNC, and Michael Pereira, currently member of Grenoble Institute of Neuroscience. So before getting started, I would like to introduce the members of the PhD committee. So first, the rapporteur, PhD reviewers. They are Simon Kelly. He will be remotely in video conference. He is a professor of cognitive science at University College Dublin. Then Bruno Rossillon, who is a directeur de recherche CNRS, director of research at CNRS at Université de Lorraine. And then the first examiner is Taryn Bolsdon, who is a research fellow, chargé de recherche CNRS in Paris. And as a professor of cognitive neuroscience at Université Grenoble-Alpes, I will be also an examiner and the chair of this PhD committee. So as you know, the PhD defense has several steps, several phases. So the first one is the presentation by the candidate of his work. After that, the jury the committee will ask questions to the candidate during about 40 minutes, maybe more, I don't know, no, maybe more, or more than 1 hour, I think, is a little bit longer. And the last step of this defense is a deliberation, reach decision and restitution to the candidate. So, François Stockart, I invite you to present your work for about 40 minutes.
Thank you very much for the introduction, and thank you to all the jury members for being here and for evaluating my work. So today I will be presenting my PhD thesis, which was entitled Electrophysiological and Computational Study of the Role of Evidence Accumulation in Perceptual Consciousness. And as you just said, Monica, I did this under the supervision of Nathan Febvre and Michael Pereira. So, great. Okay. What do I mean when I'm talking about perceptual consciousness or perceptual experience? Well, right now, you are looking at the screen, maybe you're also looking at me, and you're hearing my voice, or you're hearing some noises in the background. You might also feel some touch sensation from the seat on which you are sitting, for example. And it's not just that your brain processes this perceptual information, you also have a distinct subjective experience that comes with it. So for example, this camera, which is filming me right now, it does process information about visual information and also auditory information, but we do not think that it has a perceptual experience. Or at least most of us would agree that it doesn't. So really, what I was trying to understand is this PhD is what happens in the brain that subtends this experience that we have. So how can we study perceptual experience? Well, in the field, it has generally been operationalized by using detection or visibility reports, for example. So, for example, we ask participants to press on a button when they perceive something. And we pair this with the contrastive approach, generally, where a stimulus is presented at such an intensity that it is sometimes perceived by the participants and sometimes not. And one thing that can be done then is to look at what happens in the brain, to look at the neural correlates of consciousness, which I will call the MCC. And so this is just what happens in the brain when we perceive it versus we do not. And one example of a seminal study that looked at this would be the study by Doan and colleagues, where they used visual masking. So in one condition, they presented those words surrounded by masks that were presented early enough and late enough after the word, such that it would be perceived by the participants. So they reported that they perceived the word on almost every trial. When the masks were close enough to the word, then they would report not perceiving it. So they could compare the brain activity between those two conditions. They used two different imaging modalities. So first, fMRI. And when they looked at the activation triggered by the visible words versus masked words, they found that in the visible word condition, the activity was much stronger in temporal cortex and also stronger in frontoparietal cortex. And when they use electroencephalography, or EEG, to look at the kind of temporal decor of this response, they found that around 250 milliseconds, there was an increase in activity of the frontoparietal electrodes when participants saw the word. And this response is called the P3, or the P300. And so that was kind of the traditional approach to the NCC 20 years ago, but those measures, they do have some limits. So the problem with fMRI, for example, is that it has an excellent or very good spatial resolution, but not such a good temporal resolution. And EEG has the reverse issue. It has very good temporal resolution, but not such a good spatial resolution. And theories of consciousness make predictions that are quite fine in a spatio-temporal fashion. So they make predictions about how when certain brain regions respond to stimuli. But one method that we have in humans to look at brain activity with both a good spatial and temporal resolution is intracranial EEG. And so this method is generally used on patients with epilepsy, so it's very invasive. The reason is that those participants are being implanted for surgical screening. But what we can do is go to those participants and ask them to participate in experiments. And they either get implanted with, or in general get implanted with electrocorticography, that's less and less common, so those are electrodes on the surface of the scalp, or stereotactic EEG, SEG, with depth electrodes that go inside their brain. And then the result of this is that we can get an electrophysiological signal that we can analyze like EEG by, for example, looking at oscillations or event-related potentials, but also we get a finer spatial resolution of where the sources of the signal might be. And this approach has been used by several authors to study the MCC. So for example, this is a recent study by Fang and colleagues, where they use a different approach to perform this contrastive approach I was talking about earlier. They presented this grating at around perceptual threshold, so at such a low intensity that sometimes participants would see them, sometimes not. And then they had this smart design when they asked participants to report whether they saw the grating or not, when an answer screen was shown, but depending on the color of the fixation cross, they would have to saccade to the right or to the left to say that they saw something. So the activity that would be triggered by the grating could not just be motor preparation, because this could not be prepared before the answer screen appeared. And so they looked at channels in the prefrontal cortex of participants who performed the experiment, and they found very reliably here in red that participants who saw the gratings had this increase of activity, which was not found when participants did not see the gratings for those trials here in blue. So this activity can be reminiscent a little bit of the P3 or P300 that I showed earlier. But those studies, they also have some methodological limits. So when I was talking about the contrastive approach, basically what we are trying to do in the field when we do this is we present a stimulus, then we know that in some trials participants report that they saw the stimulus or perceived it, and other trials that they did not perceive it. And we then look at the brain activity and we say, when they perceive it, there's this brain activity, and when they don't, there isn't, so this must be the MCC. But it's not as simple as this, because there could be prerequisites to the MCC. So for example, in some trials, participants might be more attentive or have specific expectations about the stimulus. And those might actually correlate with their perception. If they're more attentive or they expect a stimulus, then they're more likely to see it or hear it. And conversely, there could also be consequences to the MCC. So activity, for example, when I perceive the stimulus, I'm going to think, oh, I saw the stimulus, I'm going to push on the button to report it. And this activity itself might be mistaken for the MCC. So that's kind of a challenge to the field to try to disentangle the MCC from those precursors and consequences. One approach that has been used to try to tackle this is the use of no-report paradigms. And so the idea here is to have two conditions where participants perceive or do not perceive the stimulus, but we do not ask them to report anything about the stimulus. So this targets specifically the consequences of the MCC. Try to take some of those consequences away to then compare what participants perceive versus do not perceive. And it has been used by several authors before. So for example, this is a fairly recent study by Cohen and colleagues, where they use a similar paradigm, so a masking paradigm, very similar to the Doan study that I presented earlier, except here they presented line drawings. And participants had to do two tasks. First, in a report condition, they had to say whether they saw a stimulus or did not. And here they reproduce the Doan, so this is one electrode over the parietal cortex, and they reproduce this result that when participants see the objects or the animal here in red, then we have this kind of positive wave, the P3 wave. But then they had another condition. And here, what they ask participants to do is, you look at the same stimulus stream, but occasionally, there will be a green circle that will appear in the sequence. And what you have to do is, during a block, count the amount of time that this green circle is presented, and ignore the rest of the stimulation. So it means that the drawings, they become completely irrelevant to the task. And then what they did is they compare those two conditions again, and they do not find evidence for the P3 wave. So this is one example of how no-report paradigms can be used to try to say, okay, this activity is linked to the consequences of the NCC and not to the NCC themselves. And this has been done with EEG and fMRI, but actually before this PhD, it had not been done in contrastive tasks with SEEG data. So during my PhD, I performed two stereotactic EEG experiments where we used no-report paradigms. The first one was entitled Cortical Evidence Accumulation for Perceptual Experience Occurs Irrespective of Report. And here we looked at this kind of ramping activity that has been described in decision-making research and tried to see if it applies to perceptual experience. And then in the second study, we looked at the neurochorus of stimulus presence and absence. I also performed some other projects on the side. So, for example, with two other members of the lab, Ramlam Shek and also Mikael, my supervisor, we wrote a journal club in the Journal of Neuroscience looking at different roles that evidence accumulation mechanisms could take in the brain. I also participated into a consensus project over best practices in unconscious processing and led this project. And I'm also working on meta-analysis of attentional stimuli generated by unconscious stimuli, attentional reflex. Sorry. But here in this PhD, I'm focusing on the first two SEEG studies, or the only two, but those two. And the first one here first, which has just come out as a preprint. So in this study, we use a visual detection paradigm where we presented faces, and we measured SEEG activity from the brain of almost 30 participants. And we focused in 4 regions of interest in particular, which had been shown to accumulate evidence and also had been linked, or were thought to maybe being linked to perceptual experience. So the first one is the ventral visual cortex, the VVC. Then we also had the superior parietal cortex, the SPC, the inferior frontal cortex, the IFC, and the dorsolateral prefrontal cortex, the DLPFC. We had quite a lot of channels for those 4 regions, but in particular in the VVC and the IFC. And in the task, what participants had to do, so they were presented with those streams of visual noise. So for 200 milliseconds, they were presented with one of those frames, which was kind of those noisy face-scrambling stimuli. And for 3 consecutive frames, so for 600 milliseconds, there was a face that was presented in this noise. And this face was presented at perceptual thresholds. Sometimes they would see it, sometimes not, at a random point in the sequence. And one advantage of this approach compared to visual masking, for example, is that the signal is less noisy because we have exactly the same stimuli for the ones that are reported as perceived versus not. In the first experiment, we asked participants to press on a button as soon as they saw a face. And here this provided us with reaction times when they pressed on the button, so in hit trials. And we also had missed trials when they did not press on a button. And so what we looked at is high gamma activity, which is thought to be a proxy of local neuronal firing. And what I'm putting it here as a function from the onset of the face stimulus. And this is data from one channel, in the or in the ventral visual cortex, I think more precisely the fusiform gyrus of one participant. And in mistrials, the activity remains somewhere around baseline after the presentation in the phase. In hit trials, on the other hand, after around 300 milliseconds, we had this increase of activity. And so channels that showed this difference between hit and mistrials, we considered responsive, and we looked at the activity in the rest of the analyses. And here, by the way, the shades are confidence intervals, and it's the case for all the 95% confidence intervals. It's the case for all the following plots. So the first question that we ask is, do neuronal populations that we pick up from with those responsive channels, do they accumulate evidence? And so, as I said earlier, when I'm talking about evidence accumulation, is this kind of ramping activity, the sampling over time of sensory evidence that has been described first and foremost in choice for perceptual decision making. But people have proposed in the past that evidence accumulation can also determine whether a stimulus is perceived or not. And in our lab, the way that we like to think about this is with the leaky evidence accumulation process. So here you have this accumulated evidence on the y-axis. So you imagine this could be thought about as a neuron or a population of neurons, but it doesn't need to be that. It's this kind of latent variable that is accumulating evidence as a function here of the time from stimulus onset again. And there's a certain threshold, which if the accumulated evidence crosses this threshold, then the stimulus becomes conscious. On the other hand, if the accumulated evidence remains below the threshold, the stimulus is not consciously perceived. And this, whether the evidence accumulated crosses the threshold, is determined by the strength of the stimulus. So the stronger the stimulus, the more evidence there is, the more likely it is to cross the threshold. But it's also determined by random noise in the system. So there might be random noise pushing it over or below the threshold. And finally, there's also a leakage factor that makes sure that this evidence is brought back after some time. So we don't like perceive things forever. At some point we stop perceiving them. And one prediction that this framework makes is that if the activity ramps faster, so for example, the high gamma activity of a population of neuron, if it ramps faster, then it should lead to faster reaction times because the threshold would have been crossed earlier. And so one way that we can visualize this, if we look at the same channel I showed earlier, would be by dividing the hit trials depending on the response times. So here we have fast trials, and we see that this kind of fast increase of activity, but in the slower trials here in dark blue, you have this kind of shallow increase. And when we look at trial by trial, trying to correlate reaction times and the slope of the high gamma activity, we find that we have a correlation for this channel. And so one thing we could look at is also over regions of interest, do hierarchical mixed effects models to see if this holds for the regions. And we actually found this effect in the VVC and the IFC, but not in the other two regions. So this is the first marker, but another thing that we can look at to try to get closer to this latent variable or try to get more general idea of what is happening in the region is to try to get something that's closer to this latent variable. And to do this, we use a multivariate decoding approach where we try to find the best discriminability possible between hit and mistrials while keeping this kind of latent variable over time. So we have this kind of time point by time point variable for each trial, but we do this by applying weights on all the channels in one region for one participant. So we have one data point per participant per region. And first we looked, okay, can we decode hit versus mistrials in this experiment above chance? And so in the VVC, at the level of the region, putting all the participants together, we do decode above chance, but it's not so good. But when we look at individual participants, we actually have 4 participants for which we can decode around 80%, or like we have an area under the curve of around 80%. But the other participants, they are around chance. And actually, we think this might just be linked to the precise implantation of the participants because they might have channels in different parts of the VVC. In the SPC, we can decode for all the participants, and in the 2 frontal regions, we could decode for around half of the participants. But the reason I introduced this is because then what we can do is we can look at, for each trial, for each hit trial, if we have a threshold, we can look at the moment at which the variable crosses this threshold. And then we can again correlate this with reaction times, assuming that if this represents the latent variable in the model or something close to it, then we should have this correlation between reaction time and the time of threshold crossing. So this is the data for this correlation for one participant, and we find that we have a significant correlation for that participant in the VVC. And if we look at where we can find those correlations in the different regions of interest, we find it in the VVC, but it's not really clear in other regions, except in the IFC, where we can find it for a lot of participants, and we also find it at the level of the region. So overall, taking those two markers, we have some evidence that the VVC and the IFC might be reflecting evidence accumulation. But I talked about evidence accumulation for perceptual experience. And this might have nothing to do with it. Participants are pressing a button. They might just be like those regions might just be accumulating evidence for a button press to prepare like a certain response. So in order to test for that, whether that was the case or whether it was linked to perceptual experience, we had two additional experiments that the same participants did. In the first experiment, they looked at the same visual stream, but they waited for the end of the trial to provide a detection response. So if they responded that they didn't see anything, it was a mistrial. And again, if they responded seeing something, it was a hit trial. And with this experiment, we do not have the immediate model preparation that participants have to do when they press immediately, but then we do not have reaction times anymore. It's kind of a trade-off. On top of that, we presented phases not only at this threshold intensity, perceptual threshold, but also at a supra-threshold intensity that was just a little bit stronger, so that participants still saw the phases in most of the trials, but sometimes would still not see it. And so what I'm plotting here is the high gamma activity from the same channel, but in this second experiment. And in missed trials at threshold intensity, the activity once again remained around baseline. But in hit trials, we again have an increase of activity. So it seems that this effect of detection is not contingent on this immediate response that participants provide. In the supra-threshold trials, we have the same pattern. So it's also not only dependent, at least, on the intensity of the stimulus. It seems that there's a main effect of detection, or well, there is a main effect of detection. And when we looked at the level of the regions, we actually found it in all the regions. So those effects of detection, regardless of immediate model response, were found quite widely in the brain. But again, this might not have something directly to do with perceptual experience. Because it's possible that participants are thinking, okay, I'm going to respond at the end of the trial. So I'm like just preparing this response later on in time. So to try to test for whether we could find responses in those regions that are not related to the task at hand, or not directly, we had a no-report experiment, where they once again looked at phases at threshold intensity or supra-threshold intensity, but here they did not have to do anything. They could just passively look at the faces and, yeah, just remain attentive, but do not do anything. And here, two things could happen. If it's just decisional activity or task-related activity in the second experiment, then we would expect that in the no-report experiment, the activity would be flat after the presentation of the faces for whatever the intensity of the stimulus. But if there's something here to do about the perceptual experience, then we would actually expect to find more activity in supra-threshold and threshold trials. The reason is simply because when the intensity is stronger, participants should see the faces more often, a bit like this effect of detection in the second experiment. And so what do we find when we look at the same channel, where in threshold trials, the activity maybe increases a little bit, but it's not very clear. But in supra-threshold trials, we have more activity. So it seems that this channel is doing something beyond just responding to a task. And when we look at the regions of interest, we find that this effect can be found over the whole VVC, but not in the other regions. So it seems to be quite more specific than the detection effect that we found in the second experiment. So, kind of wrapping up the reasoning behind this. Here I'm showing data for the whole VVC, averaging the data across all the responsive channels in this region. So we find evidence for evidence accumulation, or like that this region might be reflecting evidence accumulation in the first experiment. Then we find evidence that it reflects detection free when there isn't an immediate motor response. And then in a no-report experiment, we found that there was more activity in suprthreshold and threshold trials that somehow mirrored this effect of detection. So what we kind of want to argue is that this region might be implementing evidence accumulation for perceptual experience. But it could also be that different neurons might be doing different, or like different populations in the VVC might be doing or implementing those different mechanisms. So to try to test whether that was the case, whether the same neural code was accumulating evidence and also responding to stimulus intensity, we use a multivariate decoding approach. So using this decoder that I introduced earlier. And what we first did is we tried to see whether we could decode hit versus miss trials in the second experiment based on the first experiment. And so that was the case. So actually the activity from the first experiment generalized to the second one. But then more crucially, we wanted to see whether we could decode the intensity on the stimulus based on whether the stimulus was being perceived, so hit versus miss trials, in the first experiment. Again, having this kind of thought process of like, in the suprartificial trials, they're more likely to see it. So if it's the same activity, we should find some effect of decoding. And we did, although I have to say that we have more limited data in the third experiment, 4 participants that showed significant decoding, but still we found significant activity for 1 participant, and then when we looked at the 2 participants for which we had those data, we found that it was above chance with those data together. Additionally, we thought, okay, this model, can we just do model simulations that would reproduce some of the results that we find in the VVC? And this, I have to say, this is mostly the work of another PhD from the lab, Ramam Shek, who has done a lot of computational work on behavioral data as well. And then we thought, can we then reproduce those traces? And we can. And so it's important to say that this model, it was fit to the behavioral data, but it had never seen the neural data. And so with some reasonable parameters, we could reproduce some idiosyncratic features of the data in all 3 experiments. So kind of wrapping up this first study, what we were looking at here was whether we could find neural correlates of evidence accumulation for perceptual experience, and we find some evidence that this might be done in the VVC. Interestingly, we find widespread evidence accumulation in the brain, so not only in our regions of interest, but we also find other regions that, well, seem to reflect those markers of evidence accumulation. But on the other hand, we find that very few regions show an effect of intensity in no-report experiments. So beyond the VVC, we only found it in the lateral visual cortex and also in some isolated channels with a cluster in the anterior insula. One thing that's quite surprising with our data is that we find late detection effects in the VVC. We might have a chance to talk about it more later, but one thing that we thought is that this could be caused by the kind of stimuli that we used being presented at perceptual threshold. And 2 other features about the data that I want to talk about is that we could actually decode false alarms. So in the VVC, we found some evidence that there was a representation of faces that were represented, that were reported by participants to be seen when no face was presented. And then also, we tested another feature of the model that I haven't talked about so far, which is the representation of perceptual confidence. So the model predicts that perceptual confidence is the difference between the maximal evidence accumulated and the threshold value. And we find some evidence of an encoding of this in the VVC. And that's also something that is quite interesting about this framework, is that it's the goal is to kind of try to explain other things than just seen versus unseen. And so in the second experiment, we actually wanted to test some further features that we could get from the model, which is first the neural correlates of absence, but also the temporal dynamics of perceptual experience. And so the main question that we ask here is, how does the brain represent the absence of a content? And when I talk about representation of absence, I do not mean absence of experience. It's not like someone being asleep or someone not paying attention, so not seeing something. What I mean is you see or you hear that something is not there. So for example, here you have kind of an experience of this egg being absent. Or another example is you are home and you hear your refrigerator compressor making noise. and at some point it stops, and you really hear something, like you hear the absence, the silence. And there's actually some psychological evidence that those types of absences can be processed as a distinct perceptual content. And there's also neurons in the brain that encode such absences. And so an interesting question is to try to see whether we can find signatures of content absence and how those kind of relate to signatures of content presence. And to do this, we used, again, stereotactic EEG. We have data from 13 patients, and we had this continuous auditory paradigm that sounded like this. So this is a babble noise stimulus. So you hear kind of those people talking, but you can't make out what they're saying. And we use those stimuli in a continuous block for 2 minutes, where we modulated the intensity of the stimulus to either be no sound, so no sound is being played, or at the threshold intensity, participants would sometimes hear it, sometimes not. So again, perceptual threshold, or hear an intensity that was so strong that we would basically perceive it all the time. And we had 3 types of blocks. We had report sound blocks, when we asked participants to press on the button for as long as they heard a sound. So press when they hear and release when they stop hearing. And a report silence block, where we reversed the motor contingency and asked them to press for as long as they heard a silence and release when they hear a sound. Finally, we had no report box, where we just asked them to remain attentive to the sounds, but do not press when you hear or don't hear something. And so, as a first approximation of what's happening here, we wanted to look at responses to sounds and silences of the brain, in the brain. So, irrespective of what participants do, so across all those blocks and irrespective of their responses. And so, this is a visualization of the channels in the brain. And in green, you have the sound-sensitive channels. And those are the channels that modulate the high gamma activity when the sound goes from no sound to a sound that they clearly hear. In orange, we have silence responsive channels that modulate the intensity, modulate the activity when it goes from a supra-threshold sound to no sound. In blue, we have channels that show both types of activities, so that responded to both sounds and silences, and black channels that showed neither. And so one way that I've represented this is by looking at the t-value. So here, each point is one individual channel. And so this is just a space determined by the t-value of the sound responsivity on the x-axis, so how strongly they respond to sounds, and on the y-axis, how strongly they respond to silences. And here I want to just focus on 2 types of populations that responded to both. So this first type of channels increase its activity in response to sounds and decrease it in response to silences, and is mostly found in the superior temporal cortex. So this is consistent with some early sensory activity there. But then we have maybe a more interesting population that increases its activity both in response to sounds and silences. And these channels are found more widespread in the brain, so also in the temporal cortex, but also in a lot of parts of the frontal cortex. And just to give you an example of what the activity looks for those 2 types of channel, this is one channel in the superior temporal sulcus. And so when we look at the data across all blocks, this is the activity, so high gamma activity on the y-axis again, locked on the time from the transition. And the first transition I'm showing is again from no sound to supra-threshold. So there was no sound before 0, and then a sound is being played. And you just have an increase of activity. When we look at the contrary, so supra-threshold to no sound, so a silence starts, then the activity decreases. And this is just consistent with some sensory response to sounds in general. And just to have a kind of a check in the no report condition, where we ask them to do nothing, we find the same type of activity. But then this is another channel that is in the prefrontal cortex, and it shows a very different response. So it increases its activity to sounds, but also to silences. And interestingly, this is the case for this channel, but not for all the channels that show this for all blocks. But when we look at no report activity, a no report block, sorry, we also have this increase in activity. So it seems to not be linked to only reporting hearing a sound or a silence. But still, this might have nothing to do with perceptual consciousness. This might just be a prefrontal population that responds to the sensory information. So then the next thing I want to do, and I'm starting to look into, is how detection reports, so participants reporting that they hear sound or silence, how does that modulate the response? And so here, for the same channel that I was just showing the prefrontal cortex, what I'm putting here is the transition from no sound to a threshold segment, where they sometimes hear the sound, sometimes not. And here in blue, you have the activity of this channel locked on transition when they start hearing the sound, or when they say that they heard the threshold sound, versus when they say that they did not hear it. There's more activity when they heard the sound. Then we can look at another type of transition where they clearly heard the sound, and it goes to threshold intensity, and they sometimes continue hearing it, sometimes stop hearing it. And here we have the reverse pattern, and the interaction between the two types of transition is actually significant, and you can see that there's an increase of activity when they miss the sound. So when they heard the silence, when they reported hearing silence. So it looks like detection actually modulates this response that we find for this prefrontal population. So the data that I showed here, they're still somewhat preliminary. I will be working on them more in the coming months here in Grenoble. And some of the analyses that I want to do, well, first I want to look at the effect of experimental condition. So we have this condition where they report by pressing on the sound on the button when they hear a sound. This other condition when they report when hearing silence. And I want to see how this kind of changing the model contingency influences the neural activity. Also, I started to look into it. It hasn't been concluded so far, but I want to look more into whether it is possible to find evidence accumulation for sounds and for silences. I want to further characterize the high gamma activity by looking at it at the region level, for example, in the superior temporal sulcus in general, or in some specific prefrontal regions, and also the temporality of the response. So, for example, when do those detection effects start? Another thing that we want to do with this data is to kind of leverage its continuous nature to look at experience and reported duration. So participants report for the length that they hear a sound, and also the length that they hear silence. And interestingly, actually, the framework that I presented earlier, it makes predictions about experience durations, how long you perceive something. And it's the amount of time that this variable is over the threshold. And Ramam Shek, again, from the lab, she has some data from computational modeling that shows that goes in this favor. So we want to look at whether we can also find neuronal markers of that. And another thing that I haven't talked about so far is that we have an implicit measure here. So the button that we use is not all or none. We actually measure how strong participants are pressing. And we have some evidence that actually they press faster when the sound is stronger. And so what we want to do, maybe using some additional behavioral data, is to look at whether the speed of the button press could be correlated with confidence or perceived intensity, and see if this implicit measure then in turn, like we can find correlations with some neural activity. So now I'm taking a bit of a step back for the last section of this presentation, and I want to just kind of summarize and discuss some of the points I introduced earlier. So here in this PhD, I performed 2 SEEG experiments where we used no report paradigms for the first time with SEEG data, and also tried to think in a more mechanistic term about the MCC by thinking about this evidence accumulation framework. And in the first study, I looked at the cortical evidence accumulation for perceptual experience, and we found that the ventral visual cortex displays activity consistent with this. And here, perceptual experience was not only just detection, but we also looked at perceptual confidence. And in the second study, I looked at the neural correlates of stimulus presence and absence to try to investigate the representation of the absence of a content, and also in the future want to leverage this continuous paradigm to look at other aspects of perceptual experience, like experience duration or implicit confidence. So what are some of the implications of the results for the understanding that we have of perceptual experience or perceptual consciousness? Theories of consciousness, they make a lot of different kind of predictions, but 2 ways that they can be distinguished is by looking at the anatomy of the NCC, so which regions, what kind of subtend perceptual, or which networks, which subtend perceptual experience, but also the temporality of the response. So what time after a stimulus is presented does it then become represented, or does the brain then react to it? And so when we focus on the anatomy here, the result from the first study, so the phase detection study, it appears to be more consistent with NCCs being localized in the back of the brain, so not in the prefrontal or parietal cortex. So what we find here, and so this is the plot I haven't shown yet, is actually a template MNI slice, and on it we can see different channels, and the ones that are surrounded in red were found to reflect evidence accumulations. The one in blue were detection sensitive in the second experiment, and the one in purple were also intensity sensitive in the third experiment. And you can see actually that this is a cluster that's mostly localized in the mid-fusiform gyrus, kind of consistent with the location of the fusiform phase area. So those results appear kind of like compatible with the idea that some specific regions in those higher order sensory cortices might be involved in perceptual experience. But I want to kind of like mitigate this conclusion, because I think we should not really use the data to say that the prefrontal cortex plays no role in the perceptual experience. First, there's other studies that have found significant decoding of conscious contents in the prefrontal cortex. And I think maybe we might be missing also the kind of format of the activity that we should find with our decoding approach. So it has been recently suggested that the prefrontal cortex does not actually represent the contents of experience, but what it does is act more as a pointer to different representation in sensory cortices. And I think the data that we have here would not necessarily be able to kind of like extract this pointer activity. So I think the jury very much still out there for the role of those different regions in perceptual experience. Another aspect is the temporality. And as I said earlier, the results that we found in the first study, they're pretty late, like the effects of detection. And so at face value, they appear to be quite consistent with theories that say, oh, okay, consciousness happens in the brain quite late after the presentation of stimuli or after the seeing stimuli in the work. Again, I'm not sure. So I think this might also depend on the type of stimuli that are presented to participants, and the ones that we use are not super ecological. They're presented at threshold, which is not, it's not true. Most of the time when we see things, it's like the information, the evidence is very strong. Also, it is possible that the type of stimuli we use with this kind of phase scrambling, maybe participant, I do not actually think it's the case, but I think it will be interesting to discuss. They might have been, for example, received at the second frame at which the phase was presented. But it also, I think, opens the door for an interesting thought, which is that the NCC must not necessarily have a fixed temporality, which I think a lot of theories of consciousness do not take into account, and which makes sense in the framework that I presented with evidence accumulation. Because if the stimulus is very strong, you have a steep increase of evidence, and of evidence being accumulated, and then you perceive the stimulus very soon after it's presented, and the associated neuroactivity comes very soon as well. Whereas if the stimulus is very the evidence is not strong, then it can take much longer, and the effect can show up in the brain much later. So I think, yeah, this idea of temporality, maybe with this data, it would be interesting to look further if we can find different timings in the same regions, depending on the strength of the stimulus. And so this kind of links me to another question, which is the type of evidence accumulation that we look for here. And it's important to note that traditional evidence accumulation models, they do not predict what we predict here, because they predict bounded accumulation. And so here, for example, this is a study where they measured the activity of single neurons in monkeys that were reporting on the movement of those dots. So they would like just saccade to the right or to the left to report where the dots were moving, and the dots were moving with more or less strength. So there was more or less signal strength. But when you look at all those different signal strengths and you look at on the response, then the activity right before the response is always at the same level. And then there's kind of a reset and the activity goes down. But this is not what we say. And we actually wrote a this journal club article with Ramamchek and Michael Pereira, where we kind of argue that there might be different types of accumulators in the brain. And actually, in this leaky evidence accumulation process, the prediction is that after a threshold is reached, the accumulation continues, and this is what determines perceptual confidence. And other models of confidence, by the way, predicts this as well. And interestingly, studies with different modalities have actually identified different accumulator profiles, and not all of them fit the bounded accumulators model, even in decision-making. And when we look at the different potential accumulators that we have in study 1, locking the activity on the response here at 0, for example, in the VVC, we do not find the same type of response as the AFC. And so I think this is kind of going in favor also with the model stimulation results, that the VVC might be doing an accumulation that's not bounded on the threshold. So I also want to take 1 or 2 minutes to discuss the limitations of this approach. So we use stereotactic EEG signals, and those signals have some intrinsic motivation limitations, although I think the advantages that they have in terms of both spatial and temporal resolutions outweigh those limitations. Also, in the first study, we talk a lot about the ventral visual cortex as like an NCC, or playing a role in the conscious experience of faces. But I think it's important to acknowledge that it's not really expected that this is a general feature of perceptual experience. It might be quite specific to face stimuli, or at least higher order visual stimuli. And I think it's very important in the future to look for domain general NCCs within a single modality, but also across sensory modalities. Also, I want to kind of have a cautionary note on, I think, a bit of a problem sometimes with the contrastive approach. So participants sometimes report that they do not perceive something, and it's very tempting to say, oh, in this case, they were unconscious of the stimulus. And we actually have this consensus paper that is out as a preprint, and hopefully will come out soon, where we really trying to argue that people need to be careful about the phrasing that they have in the situation, because there's just different criteria in effect. They might see something, but not very clearly. So I think it's really important with those kind of data to focus more on distinction between the trials where they report seeing versus not seeing, or perceiving versus not perceiving, but not saying, oh, we have like unconscious activity. And another limitation of the contrastive approach, I think, is that as I stated in the introduction, it is not perfect at distinguishing the NCC activity from other types of activity that could correlate with it. And I think the no-report paradigms, they do provide kind of a good control for limiting the consequences of the NCC. But there's this thing called the born monkey program, which was introduced by that block, which is to say that, well, even if participants do not report anything in those no-report conditions, when they see a stimulus, they might still start to mind wander, think, oh, I remember this face, or like this face makes me think of something. And this kind of brain activity that goes with it, it will only happen when they perceive. And it's not really what we talk about when we talk about conscious perception of that stimulus. So we don't have perfect kind of, perfect disentangling of those. And also, I talked about the prerequisites of the NCC, and this, I think, in this work, but also in the field in general, has not been studied enough. And so moving to some perspectives, I think it would be very interesting in the future to study a little bit more those prerequisites to try to understand how, for example, expectations can contaminate some of the results that are interpreted as NCCs. But also one approach that I would like to kind of put forward, and I think would be very interesting, is to look at false alarms. So when participants say that they perceive something, but nothing was perceived, in this context as well, to kind of compare those intrinsic versus extrinsic perceptions and see which kind of neural activity they share and do not share, because it's interesting to look at a case where participants perceive something, but there isn't this early sensory activity because there's no stimulus out there. I think what is also a bit missing in this paper, in this work so far, is looking at other frequency bands than the high gamma activity. So there is work from some lab, including Bruno Vasson, who's here, showing that actually the similar, if not even stronger, responses to phase stimuli in the temporal cortex. And I think it would be interesting to see whether we can find evidence or markers of evidence accumulation in those other frequency bands, for example. I think it would be very interesting to go further in the kind of interactions between the regions of interest, for example, see how an evidence accumulation network could work in the brain with some functional connectivity or cortical evoked potentials, for example. I think in the future, I would like to adopt more of a also causal approach with neurostimulation. So this has been done. There's quite some results showing that stimulating the fusiform cortex can lead to distortions or hallucinations of faces. But I think there's some more precise kind of predictions that can be tested with neurostimulation. And then finally, I think the approach that we use here, it kind of completely does not look at the subcortical correlates of consciousness, because we mostly have cortical coverage. But we also know that subcortical structures play an important role in perceptual consciousness. And actually, in my postdoc at the Blumenfeld lab, which I will be studying in February, I will be looking at neurostimulation in subcorticals, well, in the thalamus in particular, in the context of perceptual experience research. So on this, I want to thank my supervisors. I want to thank all my collaborators on this project, all the patients who accepted to participate in the studies, and of course, thank the jury for evaluating.